Hello everyone. Now today we will be doing the next topic, that is urine formation. So this diagram is showing the steps of urine formation. Urine formation involves three processes. First, glomerular filtration, or we call it as ultrafiltration. Second is reabsorption, and third is tubular secretion. So three of the steps you are seeing here. Number 1, that is glomerular filtration. Number 2, that is reabsorption. Number 3, that is tubular secretion. So what is glomerular filtration? The filtration of blood takes place in the glomerulus due to capillary blood pressure, is called as glomerular filtration. And the second step, that is reabsorption. So in reabsorption, the substances like glucose, amino acids, sodium ions, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by passive transport in different segments of the nephron. From about 180 liters of filtrate formed per day, only about 1.5 liters urine is released in a day. Thus, 99% of the filtrate is reabsorbed by the renal tubules. Now, the third step is tubular secretion. Tubular secretion is an important step in the formation of urine, as it helps in the maintenance of ionic and acid-base balance of body fluid. Now here is the first step, that is glomerular filtration of urine formation. The filtration of blood takes place in the glomerulus due to capillary blood pressure through three layers. So these three layers are shown in the diagram. The endothelium of glomerular blood capillary, or we can say that the endothelium of glomerular blood vessels, then the epithelium of Bowen's capsule, and the third, a basement membrane between the two layers. The epithelial cells of Bowen's capsule, called podocytes, leave some minute spaces called filtration slits or filtration pores, through which all the constituents of the plasma except the proteins are passed into the lumen of Bowen's capsule. On an average, 1100 to 1200 milliliter of blood is filtered by the kidneys per minute. The amount of the filtrate formed by the kidney per minute is called glomerular filtration rate, or GFR. It is approximately 125 milliliter per minute, or we can say that 180 liters per day in a healthy individual. Now the second step is reabsorption. 180 liters of glomerular filtrate is produced daily, but 99% of this is reabsorbed by renal tubules. So normal volume of urine released is 1.5 liters. From the filtrate, glucose, amino acids, sodium ions, etc. are reabsorbed actively, and nitrogenous wastes are reabsorbed passively. Passive reabsorption of water occurs in the initial segments of the nephron. Third is tubular secretion. Cells of proximal convoluted tubule and distal convoluted tubule maintain ionic or sodium-potassium balance and acid-base balance of body fluids by selective secretion of hydrogen ions, potassium ions, and ammonium ions into the filtrate and absorption of HCO3 negative from it. Collecting duct maintains pH and ionic balance of blood by the secretion of hydrogen and potassium ions. So this is the NCERT diagram. This diagram is given in the NCERT book showing the different parts of the nephron, and in this the reabsorption and secretion of major substances at different parts of the nephron is shown. Now here we are discussing the functions of the tubules. Now, as you already have done the diagram and we have studied the all parts of the nephron, so first we will be discussing the functions of proximal convoluted tubule, then Hanel's loop, then distal convoluted tubule, then collecting duct. Now, functions of tubules. First we have PCT. PCT, that is proximal convoluted tubule, is lined by simple cuboidal brush-bordered epithelium which increases surface area for reabsorption. Nearly all the essential nutrients and 70 to 80% of the electrolytes and water are reabsorbed by this thin segment. Proximal convoluted tubule also helps to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ions, ammonia and potassium ions into the filtrate and by absorption of HCO3 negative ions from it. Second part is Hanel's loop or loop of Hanel. Reabsorption in this segment is minimum, but it plays a significant role in maintenance of high osmolarity of the medullary interstitial fluid. The descending limb is permeable to water and impermeable to electrolytes. This concentrates the filtrate or makes it hypertonic as it moves down. The ascending limb is impermeable to water but allows the transport of electrolytes actively or passively. Therefore, the filtrate is diluted or becomes hypotonic as it passes upward. Now the third part is DCT, distal convoluted tubule. Reabsorption of sodium ions and water takes place in this segment depending upon the condition of the body. DCT also reabsorbs HCO3 negative ions and selectively secretes hydrogen ions and potassium ions and ammonia to maintain the pH and sodium-potassium balance in the blood. Now next is collecting duct. Large amount of water could be reabsorbed from this region to produce concentrated urine. This segment also allows the passage of small amount of urea into the medullary interstitium to maintain the osmolarity. It also plays a role in maintaining pH and ionic balance of the body fluids by selective secretion of potassium ions and hydrogen ions. These functions of tubules are shown in different parts like PCT, Hanel's loop, distal convoluted tubule and collecting duct. Now these are the three steps which we have discussed in the urine formation. The first is glomerular filtration. So it is the first step of urine formation. In this step, blood enters the glomerulus via efferent arteriole. This blood is filtered in glomerulus. Water and most solutes are filtered, whereas blood proteins and blood cells are not filtered. Second is tubular reabsorption. So it is a selective process where water, glucose, amino acids, urea, ions such as potassium ions, sodium ions, chloride ions, etc. are reabsorbed. Proximal convoluted tubule reabsorbs water, sodium ions, potassium ions, glucose, amino acids, urea, bicarbonate ions, etc. Loop of Hanel absorbs water, sodium ions, glucose, amino acids, urea, calcium ions, magnesium ions, etc. are reabsorbed. DCT, reabsorption of sodium and water takes place in this segment DCT also reabsorbs bicarbonate ions and selectively secretes hydrogen and potassium ions and ammonia to maintain the pH and sodium potassium balance in the blood. Collecting duct. Large amount of water could be reabsorbed from this region to produce concentrated urine. Now this is the summary of events occurring in a nephron. So first, in the first column you are seeing material transferred. Then in the second column there is nephron region, which part of the nephron is doing what type of function. And in the third column you are seeing the process which is involved and the mechanism in the fourth column. So in first number, glucose, amino acids, vitamins, hormones, sodium ions, potassium ions, magnesium ions, calcium ions, water, urea, uric acid, creatinine, ketone bodies. These are the material transferred. The part of the nephron is Bowen's capsule. Process here involved is glomerular filtration. Mechanism is ultrafiltration. Second, glucose, amino acids, hormones, vitamins, sodium ions, potassium ions, magnesium ions, calcium ions. These are the material transferred. And the part of the nephron is PCT, proximal convoluted tubule. Process involved is reabsorption and mechanism is active transport. Now in the third number, chloride ions. The material transferred, nephron region is PCT, process involved is reabsorption, mechanism is passive transport. In the fourth part, we have water. This is the material transferred. Nephron region is PCT, process involved is reabsorption, mechanism is osmosis. Now at the fifth number, we have urea. This is the material transferred. Nephron region is PCT, process involved reabsorption, and the mechanism is diffusion. Now at sixth we have water. So this is the material transferred. The region is narrow region of descending limb of loop of Hanel. Then reabsorption. Then osmosis is the mechanism. Now seventh we have sodium ions, potassium ions, magnesium ions, calcium ions, chloride ions. The region is narrow region of ascending limb of loop of Hanel. Reabsorption is the process and the mechanism is diffusion. At eighth number, inorganic ions are there. Now the region is wide part of ascending limb of loop of Hanel. Process is reabsorption, mechanism is active transport. At ninth we have water. So the region is distal convoluted tubule, collecting duct. The process is reabsorption with ADH and ADH helps in the reabsorption. Mechanism is osmosis. Now the next is sodium ions. The region of nephron is DCT, collecting tubule, collecting duct, and the process is reabsorption with aldosterone helps reabsorption and secretion. Then the mechanism is active transport. Then urea. The region is last part of collecting duct, process, reabsorption, and it helps aldosterone, it helps reabsorption, secretion. Then mechanism is diffusion. Then we have creatinine, hippuric acid, foreign substances. These are the material transferred. The part of the nephron is PCT. Process is reabsorption with aldosterone, help reabsorption, secretion. Mechanism is active transport. Then potassium ions and hydrogen ions, the material transferred. Part of the nephron is distal convoluted tubule. Process reabsorption with aldosterone helps reabsorption, secretion. Mechanism is active transport. Then ammonia, part of the nephron involved is DCT. Process reabsorption with aldosterone help reabsorption, secretion. Mechanism is diffusion. Then urea, ascending limb of Hanel's loop, that is the thin part. Process reabsorption with aldosterone help reabsorption, secretion. Then mechanism is diffusion. Now next is the physiology of excretion. Major nitrogenous excretory substance in frog, rabbit and humans is urea. That is, these are ureotelic animals. The excretory physiology in these animals may be considered under two phases, viz. Urea synthesis and formation and excretion of urine. So synthesis of urea in liver. Urea is formed in liver by two processes, deamination then ornithine cycle. Now urine formation occurs in the kidney. It involves three processes as already we have discussed: glomerular filtration, reabsorption and tubular secretion. Now there is one mechanism of urine concentration, or we call it as counter current mechanism of urine concentration. Mammals form hypertonic urine. The urine is made hypertonic with the help of counter current multiplier system. This process takes place in the Hanel's loop and vasa recta, and it involves mainly sodium ions and chloride ions. In proximal convoluted tubule, urine is isotonic. The descending limb of loop of Hanel is permeable to water. Its surrounding tissue fluid is hypertonic. Hence, the water moves out and the sodium and chloride ions move in the descending limb by passive transport. Therefore, the filtrate in the descending limb finally becomes hypertonic. So, the transport of substance facilitated by special arrangement of Hanel's loop and vasa recta is called counter current mechanism. Now this is the diagrammatic representation of a nephron and vasa recta showing counter current mechanism. So here you are showing the diagrammatic representation of counter current mechanism. Now the flow of filtrate in the two limbs of Hanel's loop is in opposite direction and thus forms a counter current. The flow of blood through the two limbs of vasa recta is also in counter current pattern, as you are seeing in this diagram. The proximity between the Hanel's loop and vasa recta as well as the counter current in them help in maintaining increasing osmolarity towards the inner medullary interstitium, that is from 300 milliosmol per liter in the cortex to about 1200 milliosmol per liter in the inner medulla. This gradient is mainly caused by sodium chloride and urea. NaCl or sodium chloride is transported by the ascending limb of Hanel's loop, which is exchanged with the descending limb of vasa recta. NaCl or sodium chloride is returned to the interstitium by the ascending part of vasa recta. Small amount of urea enters the ascending limb of Hanel, which is transported back to interstitium by the collecting tubule. So, you are seeing in this diagram the osmolarity, that is from 300 milliosmol per liter in the cortex to about 1200 milliosmol per liter in the inner medulla.